You're listening to Conferences Online Allergy from Children's Mercy Hospital in Kansas City, Missouri. Today is September 12, 2025. We will be starting our basic immunology course starting with Sompayrac Chapter 1. This is presented by Dr. Nikita Raje. She is an Associate Professor of Internal Medicine, Pediatrics, and is the Section Chief of Allergy and Immunology at Children's Mercy Hospital in Kansas City.
Raje. I'm with Children's Mercy and University of Medical Missouri, Kansas City. Today is September 12, 2025, and we are starting our basic immunology series today. We'll be talking about Chapter 1, which is an overview from how the immune system works by Lawrence Sompayrac. We are looking at 7th edition. My email is mentioned here if you guys have any questions later on. So this is from the book. This book is really meant to give you the big picture of immune system. It goes over the rules rather than the exceptions.
Dr. Raje, I think you got muted.
Sorry. Can you hear me now? Okay.
Yeah.
Okay. So the textbook goes through the rules of the immune system. There are always going to be exceptions, but our goal with this book is to just focus on the basic rules of immunology. It goes over the current knowledge of immunology, and as you might have realized, especially the second years, that the current knowledge for immunology is a moving target and it's always evolving. And so what we know today may not be true 5 years from now, but that's what we are learning and in the process, and continue to keep an open mind of how the concepts can change as we add more new information. And then keeping in mind that immune system is made up of a lot of different players, and it is a team effort on knowing how the immune system works. We'll be using a lot of analogies to explain some of these concepts. So let's start with the first one. And as we know, the football season is starting. So thinking about the immune system like a football game, there are various players, right, on offensive side and defensive side, and keeping in mind that when you see the information about one player, it might not be the full information. For example, in a football game, if you're focused, if your screen is focused and you are watching a game on the screen, you might be looking at part of the game and may not realize why a player did take a step that you did not realize why they did that. And when you realize that some other player had another thing in mind, and you may not be seeing the full picture of that game in that screen. So same way with the immune system, if you are seeing what one of the cell is doing, it might not be the full picture, and you have to keep in mind that there are a lot of players in the immune system that might be playing a role. Thinking about the layers of immune defense, what's the first line of defense? As we know, the first line of defense is provided by the physical barriers, so skin and epithelial layers or mucous membranes. And so to give you a perspective, the skin is 2 square meters in length. So the total area covered by the skin is 2 square meters. But if you think of all the mucous membranes in the body, that's 400 square meters. That's the equivalent of 2 tennis courts that the immune system is responsible for guarding. And so the physical barriers provide that first line of defense for the overall a large area that it needs to cover. Thinking about the second line of defense, that's our innate immune system. Apart from the physical barriers, there are a lot of other parts of the innate immune system that we'll be talking about. And then the third line of defense is provided by the adaptive immune system. So let's talk about the innate immune system. So innate means that the system is naturally present. Innate immune system has been present for more than 500 million years, and it really doesn't change over a person's lifetime. So a kid is born with the innate immune system in place, and it doesn't change over time for that child, right? So to give you more information on that, think about a child walking barefoot in the yard. And if the child gets a splinter in the toe, typically that splinter has been sitting there for a while and likely will have like a virus or a bacteria on it. And so when the splinter goes into that toe, it has broken that first line of defense of physical barrier. And so the next set of defense is provided by the innate immune system. So if that splinter causes a problem, what are the typical symptoms that you're going to see? So typically you're going to see redness, swelling, pain, all the signs of inflammation. So what do you think is responsible for all these symptoms? Can you think of one cell that might be responsible for all these signs of inflammation? Anyone? Any takers?
Maybe some macrophages or white cells.
Right. Yeah.
Yeah.
Ding, ding, ding. Yeah. So that's macrophages basically responsible for all these kinds of signs of inflammation that you might be see in this patient. And so where are these cells coming from? Our cells of the immune system come from the bone marrow. Bone marrow has these self-renewing stem cells that renew the cells themselves, but also produce a bipotential cell that can become a lymphoid cell or a myeloid cell. Sorry, give me a sec. All right. So this lymphoid cells can become T cells or B cells or natural killer cells, or the myeloid cells have a lot of choice. Just like we have choice in our career, these cells can become various types of cells, different kinds of granulocytes. Right. So thinking about the macrophage, so say the first line of defense, which is the physical barrier, it's broken. The bacteria or the fungus or the virus that's on that splinter is now inside the tissue of the toe. How the macrophages are in the tissue, sitting there, looking out for anything that's coming its way. So it's just hanging out and collecting garbage. It has these, it just basically is a sentinel cell, and it's waiting to see if there is any kind of microbe that comes along that is not supposed to be in that tissue. The macrophages have these antennae that are used to recognize what are supposed to be danger molecules, and these molecules may have signs on them that are kind of saying, find me and eat me. And if the macrophages do come across these kinds of cells, these kinds of molecules, these could be, say, lipids or carbohydrates that are not typically found in humans, that give them the sign of, okay, that's an invader, and they are going to engulf that microbe. So that's the process called phagocytosis. As we know, it's going to form that pouch around that microbe, and that pouch becomes what we know as phagosome, that can then fuse with another organelle called lysosome to form a phagolysosome. This phagolysosome will have weapons to kill that microbe, so that that microbe is killed. All right, so while the macrophages can take care of most of these microbes right in the tissue, sometimes they need help. And so for that, they would have to communicate with other players of the team so that they can get help from the rest of the team members. So if there is some kind of battle going on with these microbes, the macrophages will release protein messengers that are called cytokines. And these cytokines will be released in the circulation. They'll take the message to the circulation and tell the other team players that they need help. And so the other cells can come to the site of infection and help the macrophages while the macrophages are waiting for this help to arrive, they are going to continue to fight that invader and try their best for the infection to not get worse. So the vigorous response that comes from all these immune cells that come and start fighting the infection, that response, immune response, is called inflammation. And so the changes that we see at the site of the splinter are from that inflammation. So that at that site, there are some chemicals that are given off, and there is increased blood flow to that area that causes the redness. The chemicals cause the endothelial cells to contract, and there is a space between them that's created so that there is leakage of fluid, and that causes the swelling. And then the chemicals can also stimulate the nerves and cause pain. All right, so let's talk. We talked about the macrophages. Now we move on and talk about the other team players for the innate immune system. So other cells are natural killer cells. These target cells that are infected and damaged by viruses or cancer or other microbes. There are complement proteins that are part of the innate immune system, and the goal of the complement protein is to make a membrane attack complex that can punch holes in the bacteria and kill them. We'll come back to the innate immune system in a minute, but we can move on and talk about the adaptive immune system. So the introduction to adaptive immune system to the humans came from the observation by Edward Jenner. Edward Jenner was a scientist who basically had noticed that people who are milkmaids don't get smallpox. And so what he did was he took the pus from smallpox pustules and actually introduced or injected it in a child named James Phipps. So that was a very adventurous experiment, but that was the first time that this was done, and that showed that the immune system can be trained. And because the vacca means cow, where the observation was made in terms of cowpox, the system was, or the product was, known as vaccine, and that's how we now know or how these vaccines can protect us from various infections. So the adaptive immune system adapts to the invaders over time. So innate immune system is hardwired, present right at birth, doesn't change as much during the lifetime, versus the adaptive immune system that is made up of B and T cells can adapt over time once it comes in contact with various microbes or invaders. It can change over time to make more sophisticated response. So the B cells of part of adaptive immune system are named so because they, in birds they came from Bursa fabricius. We know in humans the B cells come from the bone marrow stem cells, and they can differentiate into plasma cells that can make antibodies. Antibodies are weapons that are used against any kind of organic molecule. And these can be of different classes. They are called immunoglobulins, IgG, A, M, E. And sorry, that's a typo. The last one is IgD. How are these antibody molecules structured? They have two heavy chains, as you see in this figure, and two light chains. The part of two heavy chains and the light chain come together to form the FAB region, so the antigen binding region, and that apart from the antigen binding region, there is an FC region, which is the constant region that can bind to the receptors on different cells, such as macrophages. So the antigen binding part is important for the specificity with which it can bind to various molecules, and then the constant region is responsible for the function of the cells. The function is dependent on the class of the antigen antibody as well. So the structure of the FC region determines the class, and these classes can be various types like G, A, M, E. So the FAB region of each antibody binds to that specific antigen. So say the protein on the surface of smallpox virus, right? That small protein is going to bind to the antibody part, and the part of the antibody that's used to bind to that part of the protein, smallpox virus, is the FAB region. The antibodies with varied specificities are needed to protect against all kinds of antigens. But if you think of all kinds of antigens for which the antibodies are needed, there might be about 100 million of those antigens that against which we need antibodies. But that would use up about 10,000 heavy chains and 10,000 light chains to create those different specific FAB regions. But we know that our cells have about, say, 25,000 genes. So if B cells use up all the genes for just making those heavy chains and light chains, the B cells won't have genes to do anything else. And so we know that that system would not work. So that is the region, the FAB region structure needs a modular design for its creation. Sorry. All right, so that the diversity of the antibodies comes from the diversity in the FAB region, and that is provided by this modular design. Here, it shows that the gene segments, this is the immature B cell DNA, and the B cell DNA has various segments. These are variable segments, D segments, J segments, and the constant region segments. So these segments for the V, D, and J regions. So the V stands for variable, D for diversity, J for joining, and these segments, there are multiple of those for each of the type. So variable regions can be about 40. The segments for diversity segments is about 25, and about 6 J segments as well. So for the mature B cell DNA, there is a segment from each of these types that comes together to form the final mature DNA. And then it is bound to the constant segments for M and D. That's what makes the final B cell DNA segments that come together, and that can give rise to various antibody specificities to create those various FAB regions. All right, so say those antibodies are created, and now, typically there are various B cells with specificities after the B cells use mix and match and use those gene segments to form the heavy and light chains of antibody protein molecules. The small batch of these proteins can come together like a test batch. So there are about 3 billion B cells, but there are only few with the antibodies of one specificity. So say there are 3 billion B cells, but the B cells that are specific to make antibodies for flu vaccine or flu virus are a very few number, right? So same thing with, say, another bacteria or another virus or another fungus. So the B cells that are specific to each of the protein in that virus or that bacteria are very few. So how does the immune system select and know which ones to make more, right? So it's almost like a test batch. The antibodies that are created, the BCRs, that B cell receptors that are created, are only a few, and they are transported to the surface of the B cells and attached to the cells. And that's called BCR or B cell receptor. And those B cell receptor act as bait. They act as bait for fishing, which means that they are looking out for what they can bind to. For example, the B cell receptor that is on the surface for flu virus, if there is another bacteria that comes along, that B cell will not be able to help. But it has to be the B cell that is specific for a particular bacteria that has to come in contact with that particular B cell. So most of these BCRs are sitting there waiting for the antigen and looking out to see if they come across that. So for example, if you think of a B cell receptor that is specific for HIV virus, most people, for those people who are not going to be exposed to HIV, those B cells in them that are specific for that virus are going to be just sitting there waiting and not coming across that antigen. So the author here says that most of the BCRs just fish in vain and don't really come across that particular antigen, and their bait does not get used. But once it's selected, so say it's flu season and there is a B cell that is specific for flu virus. If once it comes across that B cell is going to divide and proliferate. And so it's going to make more of the B cells with that specificity. And so they make a group of B cells with the exact same FAB on it, or B cell receptor on its surface, to produce and that group of cells is called a clone. Once that clone is sufficiently large, it can differentiate into plasma cells, and those plasma cells will give rise to antibodies for that particular antigen. A clone can be as big as 20,000 B cells. So remember, to start with, B cell for one particular antigen like the flu virus is going to be very few number of B cells, so say 100. But then once they come across that antigen, they will make a big clone, maybe up to 20,000 B cells with the same specificity. And those B cells can then produce about 2,000 antibodies per second for that particular antigen. Once that virus or bacteria is cleared, once the invaders are conquered, these B cells, the whole clone, is going to die, leaving behind only a few. So thinking about the function of these antibodies, the antibodies have a function of identifying the invaders and then tag them for destruction. So typically, these antibodies are not the ones that are going to kill the microbe, right? They are just going to tag them for destruction. So to give you an example, at a wedding, sometimes there is a receiving line, and that receiving line is meant to meet and greet their guests, but there is also one more function that it does, is to make sure that there is no outsider that is coming in for the celebration. So if there is an outsider, the receivers might summon a bouncer to take care of that person. Same way, the antibodies are the receiving line. They are just going to look for the antigen, they're going to tag them for destruction, and then call upon other cells to take that invader out, such as macrophages. So the first function of antibody is that tagging for destruction, and that's called opsonization. Opsonization word means to prepare for eating. So the antibodies are just tagging or preparing the antigen for destruction. Once they tag that for destruction, the FC region, the tagging takes the FAB region into effect, and then the FC region of the antibody is free to bind to the phagocyte, like macrophages, that will be able to kill the microbe. Also, the viruses use cell machinery for their survival, and then kill the cell and then infect the neighboring cells. So these antibodies have another function called neutralization, where they can actually prevent the antigens from binding to that cell machinery. So they will not be able to enter the other cell and replicate, because of which that will help with preventing infection. So, or spreading the infection to other cells. So opsonization and neutralization are two major functions of the antibodies. Moving on, so that was B cells. Now we talk about T cells, and very similar process that we are going to go through for the information. So we talked about B cells, about 3 billion in number. Here we are talking about T cells. There are about 300 billion T cells. The T cells are also produced in the bone marrow, but then they go through the thymus to get trained and be selected to survive and perform their function. They also go through similar process of TCR formation that we saw in BCR formation, which means they use a modular design, and the TCRs are going to be produced by gene recombination for the V, D, and J segments coming together to give rise to the final T cell receptor. These TCRs will also bind to antigens just like the B cells, and once activated, they will proliferate to form a bigger group of T cells that are specific for that cell to form a clone. And that once the clone is selected and is big enough, it is gonna differentiate into different kinds of cells. So T cells, so B cells make antibodies once the clone is formed. T cells differentiate into different cells, such as helper T cells, killer T cells, regulatory T cells, depending on the type of microbe that we are dealing with. And the helper T cells are like the quarterback. They not only take up the information and help other cells, they also make the plan for how to go about taking care of that invader. So these are cytokine factories. The quarterbacks or the helper T cells produce cytokines to communicate with other cells and help the other cells in their job. So they may help the B cells in making antibodies, the killer T cells in killing the other infected cells. Killer T cells make sure that the infected or damaged cells undergo apoptosis. And then the regulatory T cells, once the infection is cleared, they make sure that the immune system comes back to homeostasis so that there are no extra weapons left causing overreaction or inflammation when it's not needed. So what's the difference? Why do we need both B and T cells if they're doing the same job? Well, the T cells only recognize protein antigens. B cells can recognize any organic molecule. However, the T cells only react to protein antigens. They require the protein antigens to be presented to them, whereas the B cells can recognize those antigens by themselves. So we'll see how those antigens are presented to the T cells. And once the virus is in the cell, antibodies cannot get to the virus, so it can survive in the cell, right? So the B cells can only take care of infections when they are in extracellular phase. When they are intracellular, B cells cannot reach them, and so T cells are important to take care of intracellular organisms. So those are the main differences between B and T cell immunity. Now, thinking about how the T cells need presentation of the antigen to them. So think of how there are billboards, right, that we see that present the information to us. Similarly, there are these these proteins called MHC class 1 and class 2. These proteins are on surface of various cells and they present the information to the T cells. So think about these two types of molecules, class 1 MHC and class 2 MHC, and here we show how they are different in their structure. The billboards of what is going on inside the cell. So say it's a liver cell and there is a virus in the liver cell. That virus is going to use up the machinery of the liver cell to make a lot of more viruses. So the proteins that are produced inside the liver cell are gonna be inside the cell, and the antibodies or B cells are blind to that. But these proteins are going to be presented on the surface of the cell by MHC molecule, and once those MHC molecules present the proteins that are being cooked inside the cell, the T cells are going to be alerted on what type of proteins are being produced in that liver cell. So that information that presents the antigens, the information from inside the cell, so those antigens presented on the surface are taken care of by class 1 MHC. What does that mean? Class 1 MHC is present on all nucleated cells and can present the antigens that are being made inside the cell. Class 2 MHC is on very specific cells. They are present only on professional antigen presenting cells, and those class 2 MHC molecules will present antigens that are brought to them from outside. So certain cells like macrophages or dendritic cells are called antigen presenting cells. When they ingest the antigens from outside, phagocytose and ingest the cells from outside, those proteins coming from outside are going to be presented on the surface of the cell using class 2 MHC. So class 1 MHC for intracellular proteins, class 2 MHC is for anything that's brought into the cell from outside. And these class 1 and class 2 MHC are made up of different chains. Class 1 MHC is formed by heavy chain bound to beta 2 microglobulin chain, and then class 2 MHC has one alpha chain and one beta chain. So the way the structure is different, it makes a difference in what kind of proteins can be attached to these. So class 1 MHC has a groove that has closed ends, so it can only fit a protein that is about 9 to 11 amino acids in size. However, class 2 MHC groove is larger and is open so that it can accommodate a larger peptide or protein, about 20 amino acids in length. So again, T cells need antigens, protein antigens, to be presented to them by class 1 or class 2 MHC on antigen presenting cells. While B cells can look at any kind of organic molecule, protein or non-protein, and they can directly interact with that antigen and don't need antigen presenting cells. Adaptive immune system has particular features that we want to talk about, and we'll go through each of these that are mentioned here. First one is activation. So remember that activation needs here is a T cell. T cell needs to interact with an antigen presenting cell, say a macrophage, and it presents that antigen bound to the class 2 MHC. That antigen is going to be presented to the T cell receptor, and once that interaction occurs, T cell may be ready for activation, but for them to be fully activated, it needs a second signal. And so that second signal is provided by some other molecules like B7 and CD28. So to give you an example, if you go to a bank to use your safe deposit box, you would have a key for your safe deposit box, but if you just use your key, the safety deposit box is not going to be accessed, and you need the banker to go in and use their key. And so there are two keys that are needed. It's a safety mechanism in place so that only the person who is supposed to use that safety deposit box is able to access it. Same thing with the immune system. Immune system can be has a lot of weapons that can be dangerous. And so when the immune system is activated, it is important to have a safety mechanism. So that safety mechanism is that the antigen can be presented to the TCR, that is one signal, but it needs that second signal to tell the T cell that this is a danger, this is an invader and not normal human cell that it's going to be acting against. So in general, activation of adaptive immune system needs two signals. The adaptive immune system is dependent on the secondary lymphoid organs for a lot of its function and activation. So there are, as we know, there is the lymphoid system, two plumbing systems, the cardiovascular system and the low pressure lymphatic system. Along the lymphatic system are lymph nodes, right? And the lymph nodes are where there are T cells and B cells, and it's almost like they are the workstations for the immune system. The lymph nodes are where the T or the adaptive immune system, the T and B cells are there, and they will come across antigens. And once they come across the antigens, they get activated and they start making the plan for killing that invader at that time. So in general, there are only about 100 or 1,000 T cells that are specific for a particular antigen. So how would a T cell come? The T cells are flowing through circulation or sitting in a lymph node. And how would these T cells come across an antigen that's way further, somewhere in the leg, sometimes in the brain, sometimes in the liver? How would these T cells come across those antigens? So typically the reason why we have these lymph nodes are, the example that's given is like they are the way stations, they are like the dating bars. So the antigens are flowing from, or sorry, the T cells are going from one lymph node to another looking for their date. They are looking for an antigen that they are specific for. If they come across that antigen, then they'll get activated and give this adaptive immune response. If they don't come across that antigen, they'll move on from one lymph node to another, continuing to look for the antigen that they are specific for. All right, more information about adaptive immune system. These activated cells will, once they are activated, they'll form a big clone, and they will make the weapons that they use to fight that infection, whether it's antibodies or the various types of T cells. Once the infection is cleared, the clones will die, and there are only a few T or B cells that will be left over. These are called the experienced or the memory cells, which are easier to activate, and they provide a more sophisticated and a quicker response if the same infection occurs again. Tolerance to self. So adaptive immune system has to make sure that it is only acting against invaders and not the self antigens. So B cells and T cells are pretty diverse, right? They are diverse enough to pick up, to act against any potential invader. And so they may recognize self antigens, but the B and T cells are educated to be tolerant to self. So they may come across self antigens, but they will not give rise to an immune response if they come across those self antigens. If that education fails, then you would see autoimmune diseases. All right, so this is the last slide. You're talking about the difference between innate and adaptive immune system. So think about if you go out of town and your shoes get misplaced and you need to get a new pair of shoes. You could go to a shoe shop that has various sizes and designs of shoes and pick up something that is available. Or you could go to a custom fit shop and ask for a really nice pair of shoes that you like. However, you know that it takes time for that custom fit shop to create the exact shoe that you want. But when you don't have a pair of shoes, your priority might be to just get something that you can use right away. And the same thing is with our immune system. The innate immune system is your fast fit, a system that is ready to go. It meets most of the day-to-day needs and holds the invaders at bay, while the adaptive immune system takes its time to create that sophisticated response. So that's the importance of the innate immune system. Even though it's not the most sophisticated, it is good for the day-to-day immune responses. And then the second thing is to evaluate the danger. The adaptive immune system is dangerous. It is diverse. It can provide a very sophisticated response, but adaptive immune system is blind to when it comes to knowing which antigen to react to. So if it is a self antigen or a invader, adaptive immune system is relying on the innate immune system to distinguish between those two and tell the adaptive immune system what is dangerous and what is not. So innate immune system is responsible for sensing that danger and formulating the plan. So the innate immune cells will say, where is the infection, what kind of infection it is, provide that information to the T and B cells so that they can make a game plan to and then instruct which weapons to mobilize and where should they go. And so that information, evaluation of danger, is provided by the innate immune system. So if the adaptive immune system and the T cells or helper T cells are the quarterback of the team, the dendritic cells and macrophages that provide that information to adaptive immune systems are the coach for that quarterback. So the innate immune system really plays the role of the coach for the team, even though the major play that you see is from the quarterback. All right, so that's the last slide. Questions?